Morning, my name is Daniela Navarro. I am assistant professor from Miguel Hernández University, and today I will talk about alteration of the cannabinoid system in animal experimentation and clinical pathology. The specific topic is immunohistochemistry, conventional and confocal microscopy, Western blot. We will be talking about antibodies, localization, dimerization, and colocalization, between others. The class outline will be as follows. First, we will discuss different techniques for protein, like immunohistochemistry and Western blot. The second part of this lesson, we have the endocannabinoid system. We will discuss about the characterization of the endogenous endocannabinoid system, CB1 receptor, CB2 receptor, and finally we will do the conclusions. Now, first part, we will discuss about protein detection, immunohistochemistry, and Western blot. To perform an immunohistochemistry, the tissues must be perfectly preserved with the same structure and chemical composition as in vivo. For that, we need to have a fixation on the tissue. The fixation preserves tissue morphology and composition, and we use different types of fixative like alcohol, paraformaldehyde, glutaraldehyde, between others. The second part, if you will include in paraffin wax, we need to have a dehydration to the tissue, but with paraffin wax only. The third part, we have to inclusion. You should include your piece in paraffin or agar, and then we have to cut the tissue with different devices. For example, we can use ultramicrotom and cryostat. For use that, we need to freeze the PS, and we have to cryoprotect the tissue, for example, the brain. If you would like to use the vibratome, the difference is you have to include the piece on agar, and you don't need the cryoprotect, don't need you cryoprotect your tissue. And finally, you can stain your tissue with Nisl or hematoxylinosine or immunohistochemistry. Immunohistochemistry is a specific technique that you can detect a specific protein from the cell. The immunohistochemistry technique, you have to use two different types of antibody. You know that the antibody have two types of chain. We have heavy chains and light chains in between each other. In the hypervariable heavy chain region and hypervariable light chain region, you have the antigen binding site. Then we have different types of antibodies. We have monoclonal antibodies. In this type of antibodies, recognize only one type of epitope from my protein, but the polyclonal antibodies recognize different epitopes from my protein. So we have different types of labeled antibodies. The first time we have an enzyme that it's binding to my antibody, and you put a substrate and you have a chemical reaction and you can see a stain in your tissue. Or you have electron-dense compound like ferritin to do the electron microscopy. Then you have, for example, for instance, an antibody with a fluorochrome. This fluorochrome is irradiated with a wavelength in the excitation range of the fluorochromes and to emit a light, a light of a specific color, for example, green or red or blue. The enzyme substrate reaction is based that the antibody it's bind with an enzyme. This enzyme, the name is horse-Radish peroxidase enzyme. It binds to the antibody and you put in your solution a substrate. The substrate is hydroxy peroxide and diaminobenzidine, DAB. This is our substrate, and then we produce a chemical reaction and we can see a brown precipitate. This brown precipitate I can see in my tissue. This is named conventional immunohistochemistry. We have different staining methods. We have first method is the direct method, and the first method that it was developed. We have the antigen, that is protein that I have to see, I want to see, and I have my antibody with the enzyme. That is the direct method. But in the indirect method, you have the first antibody and you have the second antibody bind with HRP enzyme. So we have two antibodies in the indirect method. So I will explain that the first antibody recognizes my antigen that is in the tissue, for example, the protein that I want to see. So I put in my solution the primary antibody, anti-X, for example, made in rabbits. That's important to know where the first antibody is synthesized, because you have to put the secondary antibody made in the same animal that you had made the first antibody. So we have the second antibody, anti-rabbit, made in goat, for example, binding with HRP enzyme or labeled HR with HRP enzyme. It's very important when I do an immunohistochemistry, when I perform an immunohistochemistry, you know what kind of block that I have to use. The kind of block depends on what type of the animal it was synthesized my secondary antibody. So I use different type of serum to block my tissue, to block the background and to see better my tissue, and I use the serum that it made the secondary antibody. For example, in this immunohistochemistry, I would like to use a goat serum, for example. Then finally we have the ABC method, avidin-biotin complex. This complex is very important to amplify my reaction. How can I do that? First, I have my antigen, I put my first antibody, I put my second antibody, but my second antibody is biotinylated. So with biotin, and you prepare a solution with avidin biotin complex and the enzyme bind or that labeled my avidin. So I have avidin biotin complex joined with my enzyme, and then I put my substrate and I see a chemical reaction. I can see a brown precipitate. So I can amplify my antigen, I can amplify my reaction. For example, in this paper in this article from our group, we did, for example, immunohistochemistry anti-neon. This antibody it's able to label mature neurons, and you can see the mature neurons in different type of animals. In the first case, you can see the vehicle animals. Here we have different fragments for the animals, and you can see the mature neurons. Here, in this part of hippocampus, we can see the pyramidal cells labeled. In the second immunohistochemistry that we published in this article, BDNF, brain-derived neural factor, this factor I use fluorochrome that emission in green in a wavelength like 488 nanometers. So I can see in green my BDNF positive cells here on the hippocampus, here on CSIC of the hippocampus, and so I can see these positive cells in this part of the hippocampus. Then I will discuss with you the Western blot analysis to detect protein, but the Western blot, you know, that is a protein identification technique. But the difference with immunohistochemistry that you can measure an relative amount of a protein references about another protein that is a protein, for example, for the cytoskeleton to the cells as actin. Proteins are separated by electrophoresis in a gel according to their molecular weight. A specific antibody, like immunohistochemistry, is used to detect a protein of interest. So finally, the result provides information about the molecular weight of the protein and its relative amount in the sample. You can look something like this, and you have the lane with the different protein with different molecular weight, and you can see your interest protein. How I should prepare my sample? The sample preparation for polyacrylamide gel electrophoresis. First, quickly dissect the tissue from the piece. For example, if I want a hippocampus from the brain, and place it on ice to avoid protein degradation. That is very important part, and I have to do very, very quickly. Then I have to freeze immediately in a liquid nitrogen and homogenize and in lysis buffer. You have to keep on mind what type of protein that I want to look, that I want to see, to detect. For that, if the protein of interest is phosphorylated protein, I have to include phosphatase inhibitors in the lysis buffer. Is very, very important, it's crucial. So, sometimes when you prepare your lysis buffer, you always use phosphorylated phosphatase inhibitor, because you can use this buffer for different type of protein, phosphorylated or not phosphorylated protein. It's very interesting to select an appropriate lysis buffer based on the cellular location of the protein of interest. Because if you have a protein on the nucleus, it's different than you have the protein on the membrane. It's too easier to look at protein or to put on a protein from the membrane than from the nucleus. Samples are usually treated with agent that disrupt the three-dimensional structure of protein, dimers, tertiary structure, etc. The loading buffer contains SDS, it's sodium dodecyl sulfate, and mercaptoethanol or DTT, dithiotrietol, because the CDS denature the protein and in its primary structure and coat it with negative charge. This is very important. Then we have mercaptoethanol and DDT are reducing agent that break disulfide bonds. So we have a detergent like SDZ that denature the protein, and we have mercaptoethanol that break disulfide bond. But pay attention, because some antibodies can only bind to native protein and not to denatured protein. SDS and mercaptoethanol cannot be used. This allows the protein to be separated on a molecular weight basis by SDS page. Reduction and denaturation also allow the antibody to access its binding site. What is SDS page? It's sodium dodecyl sulfate page polyacrylamide gel electrophoresis. The polyacrylamide gel is placed in a compartment containing a migration buffer, like here. You have your gel that you have to put in a buffer. Protein lysate are loaded onto the lanes because you have different lanes of the gel. You have one lane for the different protein with different molecular weight, and in different lane you can put your lysate protein. An electric file is applied, like here in this picture. In the top, an electric file is applied which causes the protein to move toward the positive pole, because we have to remember that the proteins are negative charged. Since the proteins are treated with HDAs, negative charge, the endogenous charge are negligible. The proteins are separated according to their molecular weight, so the smaller protein migrate faster. So, after electrophoresis, the protein of interest must be labeled to visualize it. What had to do? We had to do a detection and immunohistochemistry. The second part of Western blot is similar to an immunohistochemistry. So the gel is not suitable surface for labeling proteins. Proteins must be transferred to a more suitable support. I have a membrane that I can do my immunohistochemistry on it. Negative charge protein migrate to the positive pole under the influence of an electric field. For example, you have here negative pole and positive pole. You put sponge, different layer of filter paper, you have your gel, your membrane, and different filter paper, sponge, and positive pole. The protein immigration is from negative pole to positive pole. And you can see the proteins are separated here in my gel. I can see it the same in my membrane. The proteins adhere to the membrane in the same arrangement as in the gel. To do my immunodetection, first I have my membrane. I have to blockage my membrane with, for example, milk or different type of serum. Then I have to incubation with the primary antibody, wash, incubation with the secondary antibody as an immunohistochemistry, but the secondary antibody is conjugate. Then I have to wash, then I have to detect. A chemiluminescent substrate such as ACL reacts with a secondary antibody conjugate with my enzyme, that HRP enzyme, producing a signal that is captured on photographic film. Nowadays, I can use fluorochrome labeled or conjugate to my secondary antibody. I can see my membrane in a fluorescent microscope. So, for example, this is my example. I have this protein kinase 4, molecular weight 53, and the first antibody was made in rabbit. I put a lane with different protein with different molecular weight. I know that my protein molecular weight is 53, and you can see my protein here, and I can see the integrative protein beta-actin that I have to comparison this with this because it's a relative measure. So take a look. Now we will take a look to endocannabinoid system. will we discuss about characterization of the endogenous endocannabinoid system. We will talking about receptor 1 and receptor 2 a little bit, and then I will give you a lot of many conclusions. So then the endocannabinoid system, as you know, is compounded by the different endocannabinoids like anandamide that was isolated in Mechoulam laboratories in the 60s. Then they found the 2-arachidonyl glycerol, and both in the brain, the 2-arachidonyl glycerol is in higher proportion than anandamide. Both act on CB1 and CB2 receptor, and we have the cannabis sativa indica, that is my plant that I can extract the natural cannabinoids like delta 9 THC or delta 8 THC or cannabidiol or cannabinol. So we have different types of endocannabinoids like anandamide, 2-arachidonyl glycerol, but we have more. We have noladied ether, NADA, N-arachidonyl, dopamine, DIA, PIA, etc. But we have synthetic cannabinoid too. For example, agonists from CB1 receptor like CP55-19-4LO or Win55 to 212, or agonist to receptor 2 like GWH 133, or antagonist for CB1 or CB2. You know that Win 55 to 112 it's it's a full agonist at CB1 cannabinoid receptor and has a much higher affinity than THC. And this one, the antagonist rimonabant SR1415-16, it was withdrawn to the market because they have a little adverse effect. So the cannabinoid receptor are in presynaptic part of the synapses, and when the cannabinoid receptor CB1 or CB2 is activated but an agonist, they block adenyl cyclase and block the protein kinase, and then preventing the neurotransmitter release because block the gene expression. So the activation of the receptor blocks the calcium entrance to the cell. So if this terminal, if GABAergic terminal, this terminal, when cannabinoid receptor are activated, can release GABA. So when the natural cannabinoids or endocannabinoids or synthetic cannabinoids bind my receptor, it activates a G protein that activates adenylato cyclase adenylyl cyclase enzyme, and this enzyme blocks the synthesis the AMP cyclic. So my neurotransmitter is blocked. So the cannabinoid system is a neuromodulator system. When anandamide acts on the receptor, is transported into the cell through different specific transporters, and then its degradation is with the specific enzyme Fa. This enzyme degrades anandamide in arachidonic acid and ethanolamine. And then we have 2-arachidonyl glycerol, 2-AG, acts on CB1 and CB2. It's transported inside the cells through the specific transporter and degraded with the monoacylglycerol lipase in arachidonic acid and glycerol. So how can what can I do if I need more anandamide in the synaptic space? So anandamide is released and it's bind or act on CB1 receptor that activates a G protein and inhibit a calcium channel. Then the anandamide is transported into the cell through the specific transporter, and it's degraded with a fatty acid enzyme in ethanolamine and arachidonic acid. So if I want more anandamide in the synaptic space, I need inhibit its degradation. So I have specific inhibitor to the my transporter or the specific inhibitor of the degradation enzyme. So if this specific inhibitor make that I have more anandamide in the synaptic space. Now we will study the G protein linking receptor, CMP signaling. I have my receptor, GPCR receptor, and when my CD1 or CD2 act on this receptor activate a G protein. The alpha subunit of G protein activate one enzyme that named adenylate cyclase. This enzyme transforms ATP in AP cyclic. AP cyclics bind protein kinase A, and the protein kinase A go to the nucleus, and in the nucleus can phosphorylate transcription factor CREB and activate this transcription factor and start the transcription. So I have different hormones or endocannabinoids or cannabinoids, natural cannabinoids, binds to this receptor, active G protein, active adenylate cyclase, active protein kinase, and active PrEP. In case of cannabinoid receptor, if this is where it will be a cannabinoid receptor, when the cannabinoid receptor the endocannabinoid bind to the cannabinoid receptor, this pathway is inhibited. So I haven't PrEP and have a change in gene expression. I have a neurotransmitter. So where I can find the cannabinoid receptor? I know the cannabinoid receptor are in the brain, but they are on heart, on kidney, in the gastrointestinal system, pancreas, muscle, bone, immune system, kidney, lungs. So where I can find the cannabinoid receptor is its action. Cannabinoid receptor CB1 or CB2, expression and function. In the brain, I can find the cannabinoid receptor in the basal ganglia and cerebellum, so I can control of motor activity. But I can find the cannabinoid receptor on hippocampus and cerebral cortex and can act on memory and cognition. If I can find my cannabinoid receptor on amygdala, they can act on emotion. In thalamus, in sensory perception, in hypothalamus, pons, and medulla, autonomic and endocrine function. I can find my cannabinoid receptor type 1 in peripheral nerves terminal, extraneuronal areas, testicles, eyes, vascular endothelium, and spleen. The CB2 first. The CB2 receptor are in immune system, in T cells, B cells, lymphocytes, macrophages, tissue, spleen, lymphatic nodes. In central nervous system, I can act on the CB2 receptor and I can modulate the stress, anxiety, psychosis, addiction, and memory. Process of neurogenesis and synaptic plasticity. First, we will discuss about the CB1 receptor distribution. We can see that the CB1 its distribution is high in GABAergic synaptic terminals, low in glutamatergic synaptic terminals, and very, very low in astrocyte. CB1 receptor, it was found in the mitochondria of neuron and astrocyte, but it was found scattered labeling of CB1 in the rest of microglia. And then we will talk about in human microglia. Then, where are the CB1 and CB2 receptor? For example, here in the ventral tegmental area, we can find the soma of dopaminergic neuron. We can find GABAergic and glutamatergic neuron. that act on dopaminergic neuron. In nucleus accumbens, we can find the inhibitory neurons and the projection from the prefrontal cortex. So, in BTA, ventral tegmental area, GABAergic neurons here, and glutamatergic neurons, terminal, exposed to dendrite to dopaminergic neuron that can modulate the release of GABA and glutamate. And then in dopaminergic neuron 2 and in the nucleus accumbens, we can find the CB1 receptor in inhibitory neurons, GABAergic neurons, parvalbumin positive neurons, for example, and in the excitatory terminals from the prefrontal cortex. But I can see the CB1 receptor in dopamine terminals. And the CB2 receptor are in the dendrite of dopaminergic neurons and non-dopaminergic neurons, whereas its stimulation induce neuronal inhibition and reduce dopamine release in the nucleus accumbens. The CB2 activation modulates the dopamine or reduce dopamine release on the nucleus accumbens. Cannabinoid receptors. First, I would like to tell you an history about the cannabinoid receptor. First, CB1 receptor, the characterization of this receptor in the brain for the first time, and then we will discuss about the CB2 receptor and the characterization in the peripheral and in the central nervous system. In 1990, Herkenham, it was found that CB1 receptor, this is a radiolabeling area related to cognition and movement. The CB1 receptor is expressed in areas that release to cognition, like hippocampus or prefrontal cortex, or for example movement like caudat putamen, globo pallidus, or substantia nigra, or cerebellum. In the 90s, it was already identified, this receptor. Then in 1993, Matsuda, this work from Matsuda, found that by in situ hybridation, a uniform labeling in the hypothalamus, thalamus, basal ganglia, cerebellum, and brainstem of the cannabinoid receptor 1. And finally, in 23, this article from Harkani can, by immunohistochemistry, identify the different region where I can see the cannabinoid receptor and the glutamate transporter 3 overload. So I want to tell you that in 2003, it was already characterized the CB1 receptor in the brain, for example, in the rodent brain, in this case in rats. In conclusion, the localization of CB1 receptor mRNA suggests that sensory, motor, cognitive, limbic, and autonomic systems are all affected by activation of this receptor. Because remember that we found this receptor in globus pallidus, substantia nigra, hypothalamus, prefrontal cortex. So the CB1 receptor was found mainly in axon and terminals. For example, high levels of CB1 receptor are present in the stratonigral pathway and substantia nigra, but nigral neurons do not express CB1 mRNA. This finding suggests that CB1 receptors are synthesized in striatal projection neurons and transported to their axon. The axonal and terminal localization of CB1 receptor together with the observation that CB1 receptor inhibit presynaptic calcium channels suggest that CB1 receptor inhibit the release of other neurotransmitters. It's a neuromodulator system that I told you before. That was the conclusion when it was observed the expression of this receptor on the brain. The second story I would like to tell you it's about CB2 characterization. The CB2 characterization it was for the first time in 1993, but with Munro saw this receptor, this expression in the marginal zone of the spleen, but Munro, he can see this receptor on the rat brain. He made an autoradiography of an in situ hybridation. So, for the first time, it was characterized to the CB2 receptor, but they thought that it was a peripheral receptor. But in 2003, this article from Benito, from the Romero group, saw that the cannabinoid receptor 2 are selective overexpressive in neuritic plaque associated to glia in Alzheimer's disease of the human. So, in this case, by immunohistochemical analysis in the hippocampus and entorhinal cortex of the patient with Alzheimer's disease, they saw that CB2 is expressed in astrocytes associated with the neuritic plaque, whereas cannabinoid 1 receptor expression remains unaltered. So they saw that cannabinoid receptor 2 can be postulated as a possible modulator of the inflammatory response associated with this neurodegenerative process. That is important, that they saw that it was the only expression of the cannabinoid receptor 2, it was when you have a neurodegenerative process, and as possible target for new therapeutic approach. But this work from 2005 from Van Sicklen, Marha Van Sicklen, it's very, very important work because she saw she can characterize the CD2 receptor, the expression of CD2 receptor in the brainstem in non-pathological condition. Open up the possibility of non-psychotropic therapeutic intervention. So in this article, I can see the CD2 in mouse, in wild-type knockout of CD2, in green, in ferret, and in rat, the neuron, and the merge. I can see that the CD2 receptor is expressed in neurons, mature neurons. Immunohistochemistry, immunoreactivity for new and mature neurons, and CD2 receptor in neurons of the dorsal vagus nucleus. So, Van Sicklen could characterize this receptor in mouse, in rats, and in ferrets. She do by immunohistochemistry and Western blot 2, like can we observe here. What is the importance from this work? The importance of this work is that the presence of CD2 receptor in wild type and the expression is in neuron. So this work, very, very important work, that it was for me very important and an inflection point, can I say, that it was the characterization in 2006 of CD2 receptor at the brain level in the rats. This work from Jumping Gone, from the George Uhl group, they use 3 different types of antibodies. They use Cayman antibody that identified different epitope from amino acid 2033, Sigma antibody, and Santa Cruz antibody. And they do Western blot 2. Here in this picture we can see, for example, in C and T Cayman antibody, this is cerebellum, and we can see Sigma antibody. Diagram showing the localization of CD2 epitope of 3 different antibodies for Cayman, Sigma, and Santa Cruz. Okay, so they conclude the immunostain of CD2 using Cayman receptor, Cayman antibody in C2 of the hippocampus. We can see an intense staining in pyramidal cells. This is a cell culture of cell that express cannabinoid CD2 receptor, and here we can see in the spleen and in the brain of mice. Here we have cell culture and hippocampus with pre-absorbed antibody. So here we can see that the cannabinoid receptor is expressed in the brain of the rat in non-pathological condition. So the importance from this work was the different antibody used to identify CV2 receptor. They look at abundant immunohistochemistry for CV2 was observed in neurons in various areas of the right brain, for example, caudado putamen, cerebral cortex, hippocampus. The expression of CV2 receptor in glial and neuronal pattern in various brain regions suggests a re-evaluation of the possible roles that CV2 might play in the brain. That is a schematic representation of the brain distribution of the CV2. We have neural bodies in dots and axon in cruxes. So this was a fantastic characterization about the expression of CV2 receptor. This article from the same group, the UL group, in 2007, the CV2 receptor is positive in apical dendrite. Here, look at here, where is the, here, in the apical dendrite and in the cell bodies of pyramidal neurons of rat's cerebral cortex and in mouse cerebral cortex. And this group, the Alicia Brusco group, made this electron microscope, and the CV2 receptor expressed in dendrites received multiple synaptic contact from axon terminal lucky CV2 receptor, and a CV2 positive cell dendrite was contacted by non-immunoreactive axon terminal. For that staining in dendrites and cell body, dendrites, and in cell body indicate postsynaptic localization in the areas analyzed, hippocampus and cerebral cortex. So, in the first works from this group, they made a wonderful identification from the expression of CV2 receptor, and in this work we can see that the CV2 receptor is expressed in the presynaptic and in the postsynaptic too. Our group was able to characterize the CV2 receptor at the brain level in 2012. We did immunohistochemistry, we made immunohistochemistry in the UBTA and nuclear accumbens. We can see here in red the CV2 receptor, and you can see the CV2 receptor in the peripheral, in the cytoplasmic of the cell, and we can label mature neuron with neon antibody in green. So in the C picture, we can see the merge, and we can see in the peripheral, in the cytoplasm, the CV2 receptor expression and the nucleus in green. In the nuclear accumbens, we did the immunohistochemistry, red CV2 receptor, green mature neuron, and the same. We can see the expression of CV2 receptor on the cytoplasm, in the cytoplasm, and the nucleus in green. CV2 receptor is expressed in both neuron and astrocytes in the UBTA and nuclear accumbens part of the mesolimbic system. Here in the same work we can see the CV2 receptor in red, but we did in green GFI P, glial fibrillary protein, acidic protein. It's able to label astroglial cell, and we can see the expression of CB2 receptor on the cytoplasm of the glial cell, in both in UBTA and nucleocomponents. CB2 receptor is expressed in both neuron and astrocyte in UBTA and nucleocumben's mesolimbic system. So, another work for our group, it was this one, when we could make the immunohistochemistry. Here the CB2 receptor is in green, and we labeled the alpha 3 and alpha 4 subunit of the nicotinic cholinergic receptor. So we did double immunohistochemistry, one green, one red, and we can see that the CB2 receptor in green is expressed in alpha 3 nicotinic cholinergic receptor and in alpha 4 nicotinic cholinergic receptor. Here we have UBTA, picture A, picture B, and picture C and D we have nucleocumben's and UBTA. So we can see here in the merge that the CB2 receptor colocalized with alpha 3 and alpha 4 cholinergic subunit of the nicotinic cholinergic receptor. So the colocalization of CB2 receptor with alpha 3 and alpha 4 subunit of the nicotinic cholinergic receptor in areas of the reward system could suggest functional cooperation of both the receptor and their involvement in the regulation of nicotine effect at the mesolimbic level. This article, it was made, this article from 2018. For our group 2, we did an in situ proximity ligation assay. It's a similar technique to immunohistochemistry, but you combine with a situ hybridation. And we can see the tyrosine hydroxylase in blue, the GFP to labeled astroglial cell, CB2 receptor in red, and merge. Here we have, so CB2 receptor was found in extracellular location distinctive from dopaminergic and microglial cell in human. In human, the CB2 receptor doesn't express in microglial cell. Here another work, another article from our group. In this article we can see the expression by in situ proximity ligation assay in heteromers from CB2 and GPR55 receptor. This is, I suppose that you know what a type of this receptor is, but this receptor first was found that the researcher thought that it was the CB3 receptor, but it wasn't, because it's similar, and the endocannabinoids can act on this, but it's not part of this endocannabinoid system. So they made the merge with astroglial cell and microglial cell, and they can observe that the presence of CB2 and GPR55 receptor heteromes in both neurons and astrocytes, but not expression in microglia, in control and in suicide people. Finally, I will give you some conclusion. I would like to give you some conclusion about the endocannabinoid system, about the different techniques that we have used to characterize this system, and a little bit things about where cannabinoid receptor was found. First, the endocannabinoid system is mainly composed of CB1 and CB2 receptor. Their ligand anandamide and tyrosylglycerol, their enzyme of synthesis, NAPLG and deacylglycerol lipase, and degradation fatty acid amine hydrolase and monacylglycerol lipase. The endocannabinoid system components are widely distributed in the central nervous system and periphery, and function as a neuromodulatory system. Both CB1 receptor and CB2 receptor are found in the brain. Immunohistochemical and Western blots techniques have been used to characterize cannabinoid receptor. Although antibodies to labile CB2 receptor have been entirely effective, data published in Neuropsychopharmacology for our group confirm their presence at the brain level. The CB1 receptor and CB2 receptor are found in astroglia and neuron, but in humans, CB2 receptor is not found in microglia.